In this video, we want to consider the principles that govern the effectiveness of disinfections, and we also want to consider our first grouping of chemical disinfectants and antiseptics. Generally, chemicals are poor sterilizers. They are not terribly efficient at killing endospores. Some of them don't do a good job of killing viruses that are non-enveloped. Remember that a sterilant basically kills all forms of microbial life, so these chemicals are not effective at that. Do you know why endospores are resistant to most of the methods that we would use to kill them? Remember, endospores are resting cells. They're there for survival, and only some bacteria make them. Typically, these are soil-dwelling bacteria that may be in an environment where all the nutrients they need to survive are not constantly present. So these endospores are made in order to help those bacteria survive harsh, hard times. Typically, endospores have very little water in them, which makes them resistant to heat damage. Typically, these endospores have very thick protein coats that help them resist chemicals like disinfectants and antiseptics. Typically, these are resistant to desiccation, again, because they don't have a lot of water to start out with. And then these endospores are also resistant to UV light damage because their DNA is bent around special proteins that help minimize the likelihood that a thymine dimer can be made. Most chemicals are going to be disinfectants, compounds that are aimed at destroying vegetative cells, living microorganisms. There are 4 factors that influence how effective a disinfectant or antiseptic will be. The first is the concentration of that disinfectant. Some disinfectants must be used at full strength, that is, they have to be at neat concentration. Others must be diluted to be effective. So you really need to pay attention to what concentration a chemical, disinfectant, or antiseptic has to be used at. The presence of organic matter such as proteins, lipids, etc. can actually reduce the effectiveness of some chemical disinfectants. pH is also a factor for many chemicals. They must be used at the proper pH in order to be effective. Outside of that pH, these chemicals lose their effectiveness. And one of the most important factors influencing the effectiveness of disinfection is going to be time. Most chemical disinfectants must be exposed to the bacteria for a specified amount of time in order to be effective. Anything less than that specified amount of time, and the chemicals are not that effective. For example, if you look at the back of a container of Clorox wipes or Lysol wipes, you'll see that they tell you that the Lysol or Clorox solution must be on the surface that you're trying to disinfect for somewhere between 5 and 10 minutes. At 30 seconds, you just clean the surface. Usually somewhere less than 5 minutes, you sanitize, but you really need that 5 to 10 minutes in order to really disinfect well. We can test the effectiveness of disinfectants and antiseptics a couple of different ways. One way is by using a use dilution test. In a use dilution test, metal rings or sometimes glass beads are dipped into a test bacterium and they're allowed to dry. The bacteria are allowed to dry onto the surface of the beads or the rings. These dried cultures are then placed in a disinfectant for 10 minutes at approximately room temperature, 20 degrees Celsius. The rings or beads are then transferred to sterile culture medium and incubated. If there's growth of the test bacterium in the sterile medium, then that would suggest that the disinfectant or antiseptic was not effective at eliminating the growth of microorganisms. If the culture medium remains clear, then that would suggest that the disinfectant or antiseptic is effective. So this test is basically used to determine whether or not bacteria can survive treatment or exposure to disinfectants or antiseptics. Another method that we can use to assess the effectiveness of disinfectants and antiseptics is by using a disc diffusion test. And you're already somewhat familiar with the disc diffusion test because we did it in lab. But really quickly, here you have 3 petri dishes containing solid growth medium, an agar nutrient medium. A sterile cotton swab was used to streak Staph aureus onto the leftmost plate, E. coli into the centermost plate, and then Pseudomonas aeruginosa onto the rightmost plate. The plates were allowed to sit for a few minutes, and then filter paper discs that were impregnated with 3 different disinfectants, chlorine bleach, O-phenylphenol, quaternary ammonium detergent, and hexachlorophene, were then placed onto each plate in the same positions. The presence of a zone of inhibition indicates that that chemical was effective at inhibiting the growth of the microorganisms plated onto the plate. So on the leftmost plate, you can see that chlorine bleach, O-phenylphenol, quaternary ammonium detergents, and hexachlorophene have some ability to inhibit the growth of Staph aureus, which is a gram-positive bacterium. E. coli, which is gram-negative, shows some sensitivity to chlorine bleach, O-phenylphenol, and the quaternary ammonium detergent. However, it is not sensitive to hexachlorophene. We know this because there's no zone of inhibition. The bacteria basically grew right up to the surface of the filter paper disc. Can you explain why Staph aureus might be sensitive to hexachlorophene, whereas E. coli may not be? Think about the structures of these bacteria. E. coli has an extra membrane. It has that outer membrane, which helps it resist some chemical compounds. That's why hexachlorophene shows no effectiveness against this bacterium, whereas for Staph aureus there was effective inhibition. Notice that Pseudomonas on the far right has no sensitivity to hexachlorophene, O-phenylphenol, or your quaternary ammonium detergent. This is because Pseudomonas aeruginosa is somewhat special. It is a major contributor to nosocomial infections, and it's known to be extremely resistant to many disinfectants, antiseptics, and antibiotics. During the development of this strain of bacterium, it's picked up several resistance genes that allow it to resist these chemicals. This bacterium is so resistant to some chemicals and disinfectants that it can actually grow in those chemicals. So for example, the quaternary ammonium detergent disc here shows no effectiveness against this bacterium. This bacterium is actually able to grow in that detergent. This slide basically shows you a disc diffusion test, and it illustrates the fact that concentration can be important. In this particular experiment, the disinfectant that's being used must be used at a neat concentration, that is, it must be undiluted, in order to inhibit the growth of the bacterium that's been plated on this plate. Even a 1 to 10 dilution of this disinfectant eliminates its effectivity against the bacterium. With this slide, we'll begin our discussion of various chemical compounds and their effectiveness as disinfectants and or antiseptics. If you turn to the table I gave you in class, you'll find phenols and phenolics listed under non-sterilants. These don't sterilize. Joseph Lister was the first to use phenol as a disinfectant. At the time that he was practicing medicine, it was well known that phenol was effective as a disinfectant. However, Lister figured out that if this chemical was effective at killing bacteria on inanimate surfaces, it would probably be effective at killing them on living surfaces as well. So Lister then began to use this compound as an antiseptic. In order to use phenol as an antiseptic or disinfectant, the concentration of the chemical must be greater than 1%. Here is the target for your phenols and your phenolics. The main target is the cell wall. Okay. This particular compound also has the ability to disrupt membranes. If you look at the figures at the very bottom of this slide, you'll see that phenol is basically a benzene ring, okay, which is very hydrophobic, plus one hydroxyl group, which is hydrophilic. This compound inserts itself into the cell wall and disrupts it. The chemical also has the ability to precipitate proteins. Okay, so that's your target and your mechanism. One of the benefits of using phenols and phenolics as disinfectants and antiseptics is that once you apply them to a surface, they remain active and are stable over time. So you don't need to keep reapplying these chemicals over and over and over again in short periods of time. Another benefit of using these chemical compounds is that they're active in the presence of organic matter. So you could use them to clean up or disinfect pus, saliva, fecal matter, vomit, etc. As far as effectivity, your phenols and phenolics tend to be effective against bacteria, fungi, viruses, and mycobacteria. These are not effective against endospores and non-enveloped viruses. So these are not sterilants, they are disinfectants. If you look at the figure at the bottom of the page, you see phenol on the left, which I've already described. O-phenylphenol, which you see to the right, is basically a phenol group with an extra benzene ring attached. This compound, you still find it in various disinfectants. It used to be present in Lysol, however, it is no longer in Lysol. Alright. Triclosan, like hexachlorophene, which I'll talk about in a minute, have been shown to have a cumulative and persistent effect on the skin. That is, once you apply it, it kind of exists on the skin for an extended amount of time. And continuous application, let's say you're using a soap with triclosan or hexachlorophene in it, each time that you apply that soap, you just keep adding to the strength and effectiveness of these two compounds. And your bisphenols, these are not sterilizing chemicals. Okay, none of these have been sterilants yet. Your biquinides, like your bisphenols, have a broad spectrum of activity. However, activity is pH dependent. Okay, and these compounds are not particularly effective in the presence of organic matter. These are bactericidal, however. They do directly kill bacteria, and they appear to be especially effective against gram-positive bacteria. Your example here is going to be chlorhexidine. Chlorhexidine is probably the most widely used biocide and antiseptic products. Okay, you find it in a lot of hand washes and a lot of mouth washes. Because it has broad spectrum activity, it's effective against gram-positives as well as gram-negatives. It's able to bind and adsorb or stick to soft and hard tissues. It causes very low irritation on biological tissues. Some examples of chlorhexidine containing products include Paradex, Perioguard, and Perioguard, which are both mouthwashes, Hibiclens, which is a hand wash, and Avogard, which is also a hand wash. Okay, you've probably used Hibiclens at your doctor's office, or you might have seen it at the pharmacy. Biguanides, in addition to being ineffective around a lot of organic molecules, it's ineffective against mycobacteria. Okay, endospores have some sensitivity only when these compounds are heated, when these chemicals are heated. Okay, I should note that these compounds are sporostatic. They keep endospores from germinating. Not so much the actual process of germination being stopped, but outgrowth, that is, the emergence of vegetative bacteria from the endospores. That's what is inhibited. So outgrowth, the emergence of vegetative endospores from, vegetative cells from endospores is what appears to be inhibited. Enveloped viruses are disrupted by these compounds, but non-enveloped viruses are not. What we think chlorhexidine does is that it targets the plasma membrane and changes membrane permeability. We know that this compound damages the outer layers of cells. It doesn't cause lysis or cell death by itself, not until it actually disrupts the plasma membrane. We know that once chlorhexidine passes the outer layers of the cell, it crosses the cell wall or outer membrane and then attacks the plasma membrane. So again, the target is the plasma membrane, and the mechanism is membrane disruption. This leads to leakage of the cytoplasm and cell constituents into the environment and leads to lysis and or cell death. Okay. Your next group of compounds are the halogens. Your halogens are going to include things like iodine and chlorine bleach. Iodine is rapidly bactericidal. It's effective against bacteria, including your mycobacteria. It's effective against endospores, fungi, and some viruses. What we think it does is that it alters protein synthesis and the formation of membranes by making complexes between the amino acids in the proteins and fatty acids in the membranes. We also know that iodine is capable of precipitating proteins. Precipitated proteins lose their function. They're inactive. Iodine is mainly used for disinfection and wound treatments. One disadvantage to using iodine as a disinfectant is that it stains horribly. You'll know this if you've ever had iodine applied to a wound. It stains the skin. Iodine that's used as an antiseptic typically comes in two different formulations. You have your tinctures, which are basically iodine suspended in alcohol and water, aqueous alcohol. And then you have your iodophores, which are the preferred version. In iodophores, the iodine is complexed to organic molecules and the iodine gets released slowly over time. This version of iodine is slightly less effective on fungi and endospores, but it is less irritating to living tissues than the tinctures are. Chlorine bleach is a very commonly used disinfectant, and as you now know, it's also sometimes used as an antiseptic. The active ingredient in chlorine bleach is hypochlorous acid, and there are lots of different versions of hypochlorous acid. Okay? What hypochlorous acid does is it acts as an oxidizing agent. and it inhibits the activity of enzyme systems in cells. Okay? So your target is going to be proteins. Your mechanism is that this compound oxidizes proteins. Okay. We also know that chlorine bleach can also cause the inhibition of DNA synthesis. One type of chlorine compound that you should be aware of is chloramine. Chloramine is basically a combination of chlorine plus ammonia. Okay. It's used now in a lot of different cities to treat water, okay, as part of the water purification process. The reason why it's preferred over older forms of chlorine and hypochlorous acid is because it releases chlorine over an extended period of time. Okay. It is a little bit less effective than hypochlorite, which is derived from hypochlorous acid. Excuse me. Chlorine is effective against vegetative bacteria and viruses. Chlorine bleach is the preferred disinfectant for HIV and hepatitis contaminated surfaces. As a general disinfectant, chlorine bleach is used at a 0.1% concentration. However, in the presence of organic matter, such as if you're cleaning up a blood spill or vomit or something else, you have to increase the concentration of bleach to retain its action. The concentration that's often used is 0.5%. Chlorine bleach is only effective in a pH range of 6 to 8. And what's kind of important to know about chlorine bleach as a home user as well as using it on the job, chlorine bleach's strength decreases over time on standing. So if you're having to use chlorine bleach as a disinfectant, it's far better to make it fresh every day or every time that you need to use it, rather than making it one day and then using it a couple of days later. The bleach will not be as strong. A disadvantage to using chlorine bleach as a disinfectant is at high concentration. It corrodes metal surfaces, and of course, if you've ever done laundry and gotten a bleach stain, you know that it also damages fabrics. It can cause discoloration as well as weakening the fabric. Your alcohols are next. Typically, ethanol and isopropanol are used as antiseptics and or disinfectants. Methanol is toxic and is generally not used as a antiseptic or disinfectant. The way that your alcohols work is that they denature proteins and they dissolve lipid layers. Okay? The result of this is that we're going to have interference with metabolism. Remember, for example, your electron transport chain is in the plasma membrane of the cell. So if you remove that using an alcohol, you will actually disrupt cellular respiration. Alcohols also can cause cellular lysis. One important thing to remember about your alcohols is that they must be diluted. Okay, they must be diluted. This helps the alcohol penetrate deeper into cells, okay, in order to kill them. Also, despite what our mothers did when we were young, alcohols are generally not recommended for cleaning wounds. The reason why is just as alcohols denature proteins and dissolve lipids in bacterial cells, they do the same thing to your cells when you use alcohol to clean a wound. The result can be, in some cases, a protein-like clot that's formed around the wound that bacteria can hide in and cause infection. So try not to use alcohol if you can help it. If you don't have anything better, then go for the alcohol, but generally you don't want to use it. Okay? Ethanol is effective against fungi and vegetative bacteria. It's effective against mycobacteria and some enveloped viruses. but it doesn't really have as great of an effect on your non-enveloped viruses. This is presumably due to the fact that they don't have lipids, okay, which can be dissolved. With your enveloped viruses, if you damage or remove the envelope, those viruses lose their ability to infect host cells. Non-enveloped viruses, because they have no envelope, are not as susceptible. Ethanol and isopropanol are not effective against endospores. When they're present around bacteria, they can inhibit sporulation, that is the formation of endospores. They can also inhibit germination. However, once the alcohol evaporates, those cells can go on to produce new vegetative cells. Most of the time, ethanol or isopropanol is going to be used at a 70% concentration as a disinfectant. And as an antiseptic, it may be used at a slightly lower concentration. So for example, if you look at the back of waterless hand sanitizers, okay, so those are the alcohol sanitizers. Usually the concentration of alcohol in them is somewhere around 63%. Okay. A disadvantage to using alcohols as antiseptics and disinfectants is that they can swell rubber or harden plastics, and of course they are flammable. This figure at the bottom of this slide basically shows what happens to the ability of alcohol to kill bacteria over time as a function of dilution. So in the top row, you'll see that when alcohol is used at a concentration of 100% and is exposed to bacteria, and then those bacteria are then grown on growth medium, even if the bacteria have been exposed somewhere between 10 to 50 seconds to the alcohol, they'll still grow after that exposure. When the alcohol is diluted even with 5% water, however, those bacterial cells lose their ability to grow. They are killed. I will come back with more chemicals in the next video.